Computer. There is Jeff, folks. All yours, Jeff.
Thank you, Charlie. Hello, everybody. I hope folks are staying safe and warm and dry today. It's been sleeting all morning here outside of Durham where I am, and I know that there are some folks out of power in North Carolina, so hopefully we'll be able to maintain power here throughout this presentation, and I hope everybody else is staying safe and warm. But Charlie, again, thank you for the invitation. Always a pleasure to speak to the Native Plant Society, one of my favorite organizations, since plants are one of my favorite topics. And in fact, I got my degrees with concentration in botany. And today we're going to, of course, be talking about butterflies, one of my other favorite topics. And just a nod to Lisa Delwo, who helped or who did this graphic for a logo for the program that we're looking to launch. And I'm just very pleased with this, this graphic and future potential for this program. So what I'm going to do today, here we go. Just kind of as an outline, we're going to talk about this insect apocalypse that people may have been hearing about. And we'll talk about butterflies, obviously, in particular, and their ecological significance. We'll discuss the popularity of butterfly watching or butterflying and how it can be similar to birding, similar to botanizing, looking for wildflowers and so forth. I'm going to introduce this concept of Pollard walks, a special kind of survey that can be done to monitor butterfly populations, and the potential of citizen science for looking at populations stability or increases or declines. We'll talk about Pollard Base and the North American Butterfly Network as a continental coalition of organizations looking at butterfly populations. And I want to, of course, talk about this grant proposal that I'm putting together to launch officially this project that is not yet funded. The North Carolina Butterfly Monitoring Project is a dream of mine and a few others. So talk a little bit about that. And of course, we have to look at a few common and some uncommon enjoyable butterflies, the diversity, a little bit of identification, and just have a little fun with that. Look at some of the plants that they're on as well. So again, this insect apocalypse, this concept that insect populations, both locally, regionally, and globally are in decline is garnering a lot of attention recently. It's getting attention in such major publications as New York Times Magazine, talking about this insect apocalypse and what does it mean for life on Earth. Washington Post, why are huge numbers of insects disappearing? Even Science Magazine, one of the most prestigious scientific journals in the world, big feature on insect declines, a few gains, widespread losses. So there's a lot of discussion about the fact that insect populations in general do seem to be seriously declining. If you talk to your neighbor or just look at your own observations or think about those, people are reporting that they're seeing fewer bugs around outdoor lights. Look at this next time you go to a ball game in the evening. In fact, if you look at this slide here in the lower right-hand corner, that's a photo of a street light that I took in our neighborhood in August, and there's hardly an insect around it. But I know when I was a kid looking at lights like that in the neighborhood, they were swarming with beetles and moths and things. Other observations people are making, there's fewer bugs on your car windshield when you're driving around. I remember, again, when I was a kid, my job when Dad was putting gas in the car was to get out and wash the car windshields. And they're just not needing that as much. So maybe not everything about this is bad. All right, silver linings, right? Also, every year for the last few years, people are reporting fewer butterflies that they're seeing in their yards and gardens. And also there's a lot of talk about fewer fireflies I know when I was a kid, I used to go out and collect a lot of fireflies, put them in jars, bring them inside until grandma was like, take those things back outside because they're escaping all over the house. So again, insect populations appear to be declining, but a lot of these are casual observations, which are important. They might indicate a problem, but is there really an issue? What about some hard science that actually presents some data, shows some facts? Well, one of the things that's come out of this issue of people reporting fewer insects is that in the big picture, there are precious few rigorously conducted long-term studies of insect populations. So there's not a lot of big picture data that we have that says, yes, they're in decline. Here are the numbers. What are we going to do about it, kind of thing. So it's true that there are some very important ones that have been published, some very important studies, but a lot of them are regional or localized in nature or just focused on a few species. So people, we are in some serious need of scientifically conducted long-term monitoring studies. So I got on the phone, I called up my buddy Rambo, and I said, Rambo, we got to do something. And he said, it's time to get serious. So he's on board. And that is fantastic because we need him on board, but he's still just one man. So how can we do this? How can we achieve some large scale, long-term, scientifically rigorous studies on insect populations? Well, let's take a look at what some of the problems that we're facing there. First of all, there's a huge diversity of insects, over 100,000 insect species, and experts are required to identify most individuals to species. So that's a problem. Another problem is let's face it, there's very few experts. How many people do any of us know that can identify every species of bee or fly they see in their yard? A lot of them, you've got to like capture the insect and count the number of hairs on the hind leg kind of thing. And so there's usually extensive scientific training required. There's not that many experts. These are definitely some problems that we've got in trying to get some long-term good studies on insect populations. Fortunately, we've got some solutions. I want to present some solutions of how we can approach this and how we can really get a good program going that's going to scientifically document insect populations, specifically butterflies. So the solutions that I'm going to present here, butterflies are a group of insects that we can focus on. Pollard walks is a method that we can use to do this. And how are we going to achieve this on a big scale? Citizen scientists. So let's dive into these solutions a little bit. First, we're going to focus on butterflies. Why are butterflies? Why a focus on butterflies could be a solution to this? Well, folks, there's many advantages of using butterflies for long-term studies of insect populations. Like birds, a lot of citizens, a lot of people are already interested in butterflies. They're already out watching butterflies. They're going butterflying. They're noting butterflies in their yard. They're out on wildflower walks and plant society walks, and they're noticing butterflies while they're doing that. Also, like birds, the species diversity is not overwhelming. There's a tangible number of species of butterflies that you can learn, and that is a huge advantage. North Carolina has approximately 177 species of butterflies that have been documented in the state. Compare that, there's over 475 species of birds that have been documented, and a lot of birders have a good handle on identifying most of those. Here at the Plant Society, as you know, there's about 3,000 species of native vascular plants, and a lot of folks have a good handle on at least their local wildflowers. So 177 species of butterflies, we can do this. Another advantage to using butterflies or looking at butterflies is that, like birds, like a lot of wildflowers, butterflies are relatively easy to learn to identify. And there's a lot of available resources, including some good field guides, good websites. There's organizations, clubs that can be joined. There's email lists that connect you to other knowledgeable, both amateur and professional lepidopterists that can help answer questions and provide just background as well. So there's a lot of good resources available. In fact, people often ask me what's a good butterfly book for this area if I'm getting interested in butterflies. And the two that I recommend the most, they are North American in scope. So you do have to kind of weed out the things that are in the West that are not here, but still in terms of information and photos, the Swift Guide to Butterflies of North America by Glassberg and the Kaufman Field Guide to Butterflies in North America, currently the best books, in my opinion, that I recommend for butterflying in North Carolina and of course across the continent. I mentioned websites. Harry LeGrand and Tom Howard in particular have done an amazing job of compiling and keeping up to date information about the butterflies of North Carolina, and it's all available on the state parks website on the butterflies of North Carolina. There's also a number of other private web pages with great photos and information, including Will Cook's Carolina Nature, and this Jeff Pippin guy has a website on butterflies as well. I mentioned organizations, the Carolina Butterfly Society, again, similar to the North Carolina Native Plant Society. Hey, Dennis, I saw Dennis give a wave there. Very active in founding the Carolina Butterfly Society. Great organization. There are some regional chapters. They do walks and field trips and help coordinate butterfly counts, and it's an excellent resource. I highly recommend joining them if you've got an interest in butterflies. So that's great. All right, Jeff, you're convincing me butterflies are a great thing to watch. A lot of good advantages, but what about from a science standpoint? What about butterflies from good science? Butterflies make good science sense. Butterflies, in fact, have been used in scores of ecological studies as indicator species to indicate the health or the status of habitats. Looking at habitat restoration, looking at climate change, looking at a number of different kinds of studies. They've used butterflies as the focus to help gauge the change. In fact, some landmark studies that first showed the effects of climate change on animal populations were conducted in the UK, and this was through butterfly monitoring, using butterflies as well as birds, and changes in their populations helped document, 20 some years ago, climate change is real. And a number of these publications, and these studies are getting published in top journals: Nature, Global Change Biology, Journal of Animal Ecology. And again, these are 15 to 20 years ago. And so from then to now, so many more studies have come out, and butterflies are a great model to use. And along those lines, can butterflies speak for the whole habitat? If you're monitoring butterflies, can that actually give us an idea of how the habitat, the ecosystem is doing? Well, in a word, yes. One of the reasons is, of course, butterfly caterpillars, the young butterflies, they feed on plants. And a lot of plant species, or a lot of these butterflies, the plants they feed on, they're species specific. So they only feed on certain kinds of plants, like monarchs feeding on milkweeds, for example. And of course, as we know in the Plant Society, a lot of plants are habitat specific. So therefore, if your habitat changes, the habitat degrades, for example, and some of those plant species and those plant populations start to disappear, then it makes sense that you would start to notice a decline in the butterfly population as well. So that's just another example, or one reason why butterfly population numbers can help indicate habitat change. What are some other arguments for that? Now, looking at the ecosystem services of butterflies, of course, butterflies and moths together make up the insect order of the Lepidoptera. And the Lepidoptera is the only group of insects where caterpillars, as we call them, are the larvae. And here I've got a background picture of some goldfinches on some sunflower. And everybody knows goldfinches are seed-eating birds. Finches and grosbeaks, a lot of birds eat seeds. Well, the recent studies have shown that the baby birds eat caterpillars. And even baby birds of seed-eating birds like finches, they go out and they find caterpillars to feed the babies. And so songbird populations are very reliant on caterpillars, moths, and butterflies. Other ecosystem services, other important things that butterflies and moths do, of course, are with respect to pollination. I heard Charlie talking earlier that you've got some other pollinator focus talks coming up, and that's fantastic. So in addition to being important in the food chain or the food web, Lepidoptera are important pollinators for many species of plants. And here, of course, you can see a tiger swallowtail hanging on a Turk's cap lily. And look at the underside of those swallowtail wings covered in Turk's cap lily pollen. So you know that the next flower that butterfly flies to, the next Turk's cap lily is going to have some pollen exchange as a pollinator ecosystem service that butterflies provide. All right. So now we're working on establishing butterflies are an excellent group for long-term monitoring. There's people out there that can do them, and more people can learn how pretty easily. And they speak for the habitat, but how do we do this? That takes me to our next solution, is Pollard walks. So while many scientific endeavors require expensive, specialized equipment, lots of training, Pollard walks can be conducted simply with a pair of binoculars and or a camera and or a butterfly net. Grab a clipboard with a data sheet and just a little bit of instruction, and boom, anybody can do them. This, in fact, is a shot of one of the Pollard walks that I conducted when I was working at a private ecology conservation ranch out in Montana. So it was tough, but this is one of the views I had to deal with every week in conducting some of my butterfly surveys out there. That is Pollard. Well, Ernie Pollard was a citizen science visionary. And in the 70s, he developed this method, a scientifically rigorous method that he used in the UK for surveying butterflies that volunteers could do. And this method accounted for fluctuating abundances throughout the season and sensitivity to weather, thus producing statistically analyzable results of butterfly populations. And here's a picture of Ernie walking one of the first butterfly monitoring transects in the mid-70s there in the UK. So this method has come to be known as a Pollard walk, and it is the industry standard for monitoring butterfly populations. Pollard walks are used worldwide. They have contributed significantly to hundreds of publications in the scientific literature on climate change, land use change, phenological change, and many other ecological and evolutionary studies. So how does one do a Pollard walk? Jeff, you said they're pretty simple, but really? Absolutely. It's as easy as 1, 2, 3. Step 1, decide where you want to go on your walk each week, ideally. But if you can't do it every week, that's all right. You know, if you can get a half a dozen or a dozen a year done, fantastic. More the better, of course. But just figure out where's a trail that you want to hike on a regular basis, whether it's on a local park or a local refuge or a game land. You know, all kinds of things are out there for just places you like to go for a walk. So you determine where your walk is, and then you walk it ideally once a week when it's warm enough and sunny enough at certain hours of the day. So like between 10 a.m. and 4 p.m., for example, because that's when butterflies are active. They're insects. They're most active when it's sunny and or warm. And this seems obvious, but defining the strict weather and time guidelines in the protocol was one of the innovations of actually establishing this methodology. Step 2, take a hike, go out there on your walk and just record all the butterflies that you encounter to the nearest taxonomic level that you can. This is one of the other beauties. You don't have to be an expert on every species of butterfly. There's going to be a lot of times where you're out maybe just doing a walk and this little blue butterfly flies by, and that's it. You know it was a small blue butterfly. Well, you would just grab your data sheet and you would write unidentified blue. And that is absolutely fine because you're recording it to the nearest taxonomic level that you can. And usually these surveys are conducted with binoculars, but sometimes a net can be helpful, especially if you've got a blue that flies by. Here in North Carolina, of course, there's only a few blues, generally only a couple, most places, most of the time, that you would have to sort out between an azure and a tailed blue. And that you can get a pretty good handle on that with some experience. But I say that a net can be helpful because when I was in Montana conducting my surveys, at any given time, at any given survey, there might be 9 species possible of little blue butterfly that would fly by. And believe me, when they just kind of fly by, they all look the same. So I grabbed every one that I could with a net, get a close look at it, take a picture of it, and then let it go again. And that way I could get some of them to species. But I'll tell you, they're fast. And so a lot of them just went down as unidentified blue. Then easy as 1, 2, 3, step 3, you're only recording the butterflies that you see within 5 meters of you. So just think of yourself walking in a moving 5-meter box going forward. And so if you catch something out of the corner of your eye behind you, that doesn't count on this Pollard walk per se. So this spatial restriction allows for a good statistical comparison between surveys so that you can calculate a relative population index. And this is why long-term monitoring is feasible using this simple methodology. You can look at how many did you see last year, how many 2 years ago, how many this year, because you've got this defined box and these defined times and so forth. So you're getting, scientifically analyzable data. And when you're doing your walk, you walk at a relatively steady pace, again, just looking forward. And that after you've done one, you end up with a data sheet that might look something like this. I do a Pollard walk most weeks when I can, when the weather and my schedule permits, in Duke Forest here in North Carolina, in Durham and Orange counties. And so some basic things that you want to record, of course, is your location, the date, and the personnel, so yourself. And if anyone joins you, make sure you record that too. Of course, the timing is very important. So your start time, your end time, and then some basic weather information. So, how much cloud cover was there? About 50%. You ballpark these things. How windy was it? Using the Beaufort scale, you can estimate wind. and what's the temperature. And for temperature, I just pull up my phone and look at the local, the closest weather point and grab the temperature from there. Then you go on your walk and you record the species, each species you see. And when you see one, you put a tally mark. I use a dot method rather than a slash method because it's much more space efficient. For example, if we look here at Eastern-tailed blue, in this little space, there's 10, 20, 30, there's 40, I counted there. One incidental. From this incidental column, folks, I know that if you're like me and you go on a walk and you're looking for butterflies, you don't want to totally ignore that butterfly that you see on the other side of the field. Now you can't count it as a data point because it's not in your box, but I still want to note that I saw that butterfly on my walk today. And so therefore, there's an incidental column and you can record all the species that are outside your box or the individuals in that incidental column. So not only do you have data that is rigorously scientifically obtained, but you also have like a total list of all the species you saw out on your walk. So it works out very well. Because I also enjoy dragonflies, I also monitor dragonflies. I start that list at the bottom and work up. So in this one, I had a common whitetail, blue dasher. And for those of you interested in plants, it's great and certainly not necessary, but it's great to record the plant species that you see in flower, so possible nectar sources. And so I start that down at the bottom and work up too from a space standpoint. So on this walk here that was conducted in August, you had Elephantopus and Helenium, Hypericum, Oriolaria, Prunella, Rexia, and so forth. And so now we've got a list of those. And this can also lead to some useful data for plant phenology, because if you are tallying every species of flowering, or for me, showy flower plants, potential nectar plants, and you do this on your walk year after year, and you're doing it every week, now you can start to see if there are shifts in plant flowering times and plant phenology as well. So a lot of things that you can do besides tallying the butterflies. But if you don't know your dragonflies, if you don't know your plants, that's all right. A focus on butterflies is still getting the butterfly monitoring done. So we said, hey, a solution to doing insect surveys, focus on butterflies. How are we going to do it? It's simple. Pollard walks. How are we going to achieve this over big scales? Citizen scientists. Citizen science, I want to talk about that for a minute. For the big picture, we really need spatial temporal replication of surveys at large scales over long time periods. So we want to know how butterfly populations are doing in North Carolina. We need surveys in the mountains, surveys in the Piedmont, surveys at the coast, surveys in this habitat, surveys in that habitat. The more the better, obviously. And then expand beyond North Carolina if possible too. Obviously not for the North Carolina program, but for other programs, and put that whole thing together. And you can then start to get some really good big picture answers of what's going on with insect populations at large scales. Of course, true replication means that we need academic style structured surveys. This is where Pollard walks come in, where a permanent transect or route, that trail that you decided you want to walk all the time, these permanent transects or routes are established and then just repeatedly surveyed with these similar protocols, this Pollard protocol. We do this over large geographic areas. And this is really the only way to achieve this kind of survey intensity is through citizen science. And citizen science greatly increases the power to track biodiversity patterns and processes at multiple scales. So the solutions, butterflies, powered walks, citizen science, awesome. Now, how do we put all this together? Introducing the North American Butterfly Monitoring Network. So several years ago, one of my colleagues, Leslie Reese at Georgetown University, got funded an NSF, a National Science Foundation initiative, to pull together in a coalition these independent programs that had popped up around the continent that were doing some butterfly monitoring. They had some citizen science programs, or maybe just one or two people in a few areas. And, you know, there were some folks doing this in Illinois, for example, and some folks in California or whatever, but they were all independent. There was no, you know, clearinghouse, no one place where the data were available. But this initiative is kind of pulling everything together on a continental scale. So, and this map's a couple of years old, but we've got, there are programs in California, Montana, Colorado, Texas, the Midwest, Florida, that are looking at butterflies. The eastern seaboard is really lacking, And so getting something together here in North Carolina would be really useful for the whole East and contributing to the continental picture of what's happening with insect populations and butterfly populations across the continent. There's even, it's not pictured here, but there's a couple of programs up in Ontario. And so again, pulling in the whole continent. So the overarching goal that I have, that I'm envisioning for a North Carolina butterfly monitoring program, is that the program would work with citizen scientists and existing outreach programs already in place, like, for example, with the North Carolina Botanical Gardens, the Museum of Natural Sciences in Raleigh, local Audubon chapters across the state, the new Cape Fear Bird Observatory down there in the Wilmington area, and other organizations in the mountains and across the state to establish and conduct pollard walks in various habitats across North Carolina. The data collected by the citizen scientist, the individual that goes out and does their survey, would be contributed to a national database called Pollard Base for big data scientists and statisticians to then analyze insect population trends across the country. So Pollard Base is part of this National Science Foundation initiative, and it created an internet portal database so that you could go out, run your survey, then come back, log in, choose your program, whether you're doing this in Michigan or whatever. And ideally, North Carolina will be on here at some point. Choose your program and then click on your Pollard route and enter your data. And now my computer is freezing. Got that spitting rainbow, not a good thing. Oh, sorry about that, folks.
If I still have your sharing screen,
what's that, Charlie?
I said I still have your screen up there, your sharing screen.
Right, the computer is, I've got that spinning rainbow such that it's not letting me advance a slide.
Oh, okay, I see.
So hang tight for a second.
Okay, that's fine.
Of course, I've run through this a few times over the last couple of days and I've not encountered this, so naturally we're getting it now.
Oh yeah, of course. I usually get the update to the computer at the when I'm trying to do a talk.
Exactly.
Well, we got a good look at your t-shirt right now anyway.
All right, let's try this again. See if it lets me get to where we want and can resume. All right, hopefully that's the last that does that. So again, the program that I'm proposing is to develop and direct a statewide program. Currently, I've got a grant proposal written, looking for places that I'm then taking recommendations of some foundations where they might support this.
You might have to share your screen again. We just got you.
Aha, thank you. All right, let's see if this works. Technology.
There we go.
Excellent. So again, I've got this proposal written, looking for places to submit it for funding. Basically, it would be about, I'm asking for $55,000 a year for 3 years. So a 3-year proposal, $165,000 to establish the program, get it launched, get it going, and so forth for the first 3 years of seed money. So if anyone knows of a philanthropist, a foundation, a funding agency that is interested in projects like this, obviously let me know. Full detailed proposal is available. Once we get it actually launched, then we can really work toward determining the state of North Carolina's butterfly populations. Species like monarchs. People, there's lots of interest in monarchs in recent years with reports of significant declines. Most recently, good news, some rebounds on the wintering grounds. But how are they doing specifically here in North Carolina? We can take a look at some data from the annual butterfly counts. This is data from the Durham butterfly count. And it looks like there's been some big spikes. And are monarchs declining? Are they rebounding? What's going on? The problem with data like this is these surveys are once a year, and this graph does not take into account effort. So on those high peaks, it could be there were a lot of people in great weather. On the low peaks, it could have been lousy weather and not as many folks. So how much can we really get from once a year, one spot surveys? Not great science from that kind of thing. But Pollard walks in a long-term monitoring program could seriously answer the question of how are monarchs doing across the state or in an area. And what are some other species you might see on a Pollard walk or a Native Plant Society walk looking at wildflowers? Swallowtails, several species of swallowtails, and they're always a crowd-pleasing favorite because they're big, they've got some colors, they're showy. Here's an example of some of the dark swallowtails. And one of the questions that I sometimes get is, how do you identify these things? How do you identify these things? They look alike. It can be kind of confusing. And folks, it's just like learning wildflowers. You learn some names, you learn a few field marks, you know what to look for, and you can determine which species of hepatica that is, or which species of swallowtail it is. So looking at the dark ones, let's take one quick look as an example at the orange spots on the underside of the hind wings. Here in the upper left on the pipevine swallowtail, there's one arc of round orange spots. The other two species here have 2 arcs or 2 rows of spots. So if you just see one arc, boom, pipevine, nailed it. Let's take a look to the right, spice bush swallowtail. Again, learn some names, learn some field marks. Let's look at the inner arc of orange spots. And you can see that one is missing. There's one orange spot that's been bumped out and is gone. So there's your spice bush swallowtail, great field mark for that. And looking at the bottom, at the black swallowtail, again at that inside arc of orange spots, well, it's complete. And not only is it complete, but one of the spots is doubled. So that's a great field mark diagnostic there for black swallowtail. So again, that's just one example. There's other field marks to look at, but you learn a few names, you learn a few field marks, and you are off to the races. And then when you're out, of course, you get to see incredible things, especially in the spring, like a swallowtail on the spectacular Cercis canadensis on redbud. And tiger swallowtails are the official state butterfly in North Carolina, in fact. And it's great because they're beautiful, they're showy, they're pretty common, and they nectar on a lot of different species of plants, like this Joe Pye weed. Here's some on thistle. They can even nectar down low, right next to the ground. And I think this is probably a hawkweed down there, or maybe a helenium. And then again, up high is one of the females, that darker color pattern, but you can still see the tiger stripes. Cloudless sulfur is one of the most commonly observed butterflies in the state, especially in the fall. Big numbers in some years migrate north, especially along the coast. They will often stop for nectar. And folks, cloudless sulfurs are very fond of nectaring on blue, purple, and especially red flowers. Here, of course, is on a patch of false foxglove in the genus Agolinus. Here it is on one of the pinks. Another related butterfly is the sleepy orange, enjoying some Symphyotrichum. Another group of butterflies that are a favorite and always a delight to see are the hair streaks. And juniper hair streaks are just spectacular. This juniper green coloration. They're well named because the caterpillars feed on eastern red cedar. Juniperus virginiana is the caterpillar food plant. There's even a subspecies of juniper hair streak that feeds on the subspecies of eastern red cedar along the coast. So if you're a native plant person and you like cedars and you're down there in the Cape Fear area, you've got the Juniperus silicicola or silicola, depending on how you pronounce it. And I know some people treat that as a subspecies of Juniperus virginiana. Not going to go into that. I'm not going to offer a taxonomic opinion, but it is very neat how you got two subspecies of butterflies potentially, and feeding on potentially subspecies of red cedar. Of course, one of the most popular and charismatic butterflies in North Carolina is the great purple hair streak. Spectacular combination of red and orange, black and white, blue and green. And even though they always perch with their wings closed, when they fly, the upper side is a brilliant Duke blue. Great purple hair streaks are also really cool from a plant standpoint because their caterpillars feed on mistletoes. And so a good way to look for this species of butterfly in North Carolina is to look for a large patch of mistletoes up in the trees, and then down for flowers like Macania. They love hemp vine, love Macania, down at eye level where you can find the adults nectaring. Some brush foots that you may encounter, very common across the state, include the pearl crescent and the silvery checker spot. Again, they look really similar. So how would you distinguish them? Same concept. Learn a few field marks, learn a few names. On the pearl crescent, if you look on the hind wing at the outermost row of orange spots, each one has a dark, solid center. If you look at the silvery checker spot at the outermost row of orange spots, a couple of them have a dark center that's hollow or donut shaped. And that's a great mark to separate a checker spot from a crescent. And as I promised Charlie, I threw in here a picture of a buckeye, common buckeye. And I have to say that it's a very attractive butterfly with its color patterns, its big eye spots. Here it is nectaring on Parthenium. And as a University of Michigan grad, it is very difficult to say anything positive about a buckeye. But in the fall, you can get some races of buckeyes where the underside is this gorgeous kind of brick rust red. And butterfly caterpillars in general can be difficult to spot. They often hide during the day, or they're very cryptic. But buckeye caterpillars are one of the easiest of the butterfly caterpillars to find because they're fairly showy. They're usually out during the day and they feed on different species of false foxglove. So you find a patch of Agellinus, poke around. If it's the right time of the year, you can easily see some buckeye caterpillars feeding on it. That takes us to the skippers, the last family of butterflies, a large family of butterflies. They can be challenging to identify because we don't want everything to be too easy, right? You've got three major groups: the spread wing skippers, the grass or fold wing skippers, and the giant skippers. Excuse me. Spread wing skippers usually perch with their wings flat open. The fold wings perch with their wings folded or sometimes in a jet fighter pose. And there's only a couple of species of giant skippers, and they're just incredible, very locally distributed. One of the easiest butterflies to recognize in North Carolina is actually the silver-spotted skipper, one of the spread-wing skippers. And people often ask me how butterflies get their common names, and I usually answer that it's by people who are colorblind, because the silver-spotted skipper was obviously named for its big, large white spot. In the grass skippers, these, they're kind of like the shorebirds or fall warblers of the birding world. There's a bunch of them. They can be tricky to identify. But again, one of the concepts is learning the common ones in your area, getting to know that variation. And then once you learn those well, if you see something different, figuring out what it is. Two of the most common are the sachem and the fiery skipper. The fiery skipper has some black spots that look like they were penciled in with a dark pencil. On the other hand, the sachem male has some dark smudges that look like someone just kind of tried to smudge a crayon with and make its dark marks. Now, these are just a few of the skippers. Let's look at all of them. Just kidding. This is actually only a third of the species of grass skippers in North Carolina. But the point of this slide is there's a lot of gorgeous little butterflies out there, and they're fun to learn and look for. So with that, again, just wanted to introduce you to some of the butterflies that you could be seeing on some butterfly surveys or wildflower walks or even in your backyard. And with that, I'm happy to take any questions, and thank you everybody for joining in today.
Okay, Jeff, I'm going to tell the people to put their questions in the chat, and I can read them to you or you can look at your chat. But I'm going to start off with a couple of questions of my own. One, on the Pollard walk, where you have the 5-foot cube walking around, if you have more than one person out walking, it seems like it makes sense not to have one person right behind the other. It seems like they should be spread out beside each other in some manner. Is that correct?
Well, that's a great question, Charlie. And if you are running a Pollard walk, they work best as a 1 or 2, occasionally 3 people, but more than 2 or 3 people are too many to do an actual Pollard survey. And only one person to keep it scientifically strong, only one person should be spotting the butterflies. So the second person really should be walking behind you. And if they spot a butterfly that you don't, then you can certainly put that down in the incidental column, but you wouldn't want to put it into the actual Pollard record column because they need to be single observer for the effort from a scientific standpoint of analyzing. So when I do Pollard walks, I'll bring one or two other people that are interested in going or are in training or something like that. And they generally walk more or less behind me, and generally, like I said, if they see something that I didn't see, they'll point it out. I'm like, oh yeah, I'm glad you saw that. I missed it. We'll put it over in this other column, and it works out really well. Or the other thing that people do is have that second person be the scribe or the one recording all of the data while the observer is spotting the butterflies. That's another way to do it.
Yeah, that makes sense. Would that clipboard or the data sheet, it looks like if you've already got that, would you be willing to send that to me by email, and then I could share it with people that are interested?
Absolutely, absolutely. And it's
We don't have to make it up.
Yeah, it's definitely customizable. So, excuse me. Some people run Pollard transects that have just one section. That data sheet that I showed you, the transect, the route that I walk has two. Some people divide their route into 3, 4, 5, or even more sections. Generally, you would divide it by habitat type, or if you have a long walk and you just want to like break it up into a couple of different sections from a land management standpoint or whatever, that's how you would divide up the sections. And so again, totally customizable. I am happy to send you that sheet, and anybody who is interested can tweak it, modify it. And anyone that has questions on this, let me know. Absolutely, I'm happy to help out, even though we don't have the program formally launched. If there's someone who's seriously interested in doing this or has more questions, happy to kind of point them in the right direction and encourage folks to do that until we actually get a formal program and can collect the data on a statewide basis.
I've got just two more questions, then I'll let you go to the chat. The one is, we could still, even though you don't have a program established, we could still do our walks and then enter them onto that website that you showed there. Is that correct?
Yeah, absolutely. What you would do is you would still do the walks, and you could enter the data, keep the data yourself on a Excel spreadsheet, or just keep the paper data sheets in a file until this program does get launched, and then those data can be entered online.
Okay.
In fact, one of the things, because I work on a national basis too, just part-time through my colleagues at Georgetown with this program, and we have brought on some other programs across the country that didn't know about us and we didn't know about them. We discovered each other. They've been running surveys for maybe 3 or 5 years, and then we've worked with them to get their program into Pollard Base and get the back data logged in. So that is very easy to do. In fact, like I said, I'm running these Duke Forest surveys. I've done that for about 3 years, but nothing's entered into Pollard Base yet because this program hasn't been launched and funded from the North Carolina standpoint.
Okay, my last question then on that is, would you be willing to do a workshop later on, like in the summer or whenever you get funded, if we want our members to learn how to do it exactly,
Absolutely. Like I said, one of my goals is getting this funded, and then I would travel across the state working with different groups and giving workshops and helping folks get started doing whatever necessary. So yeah, we'd definitely love to do that.
Okay, well then I'll turn it over to you if you want to read the chats there and answer the questions.
Well, is it better for me to do that or for you to kind of moderate? If you read,
I can do it either way. I can open up the chat and,
how about if you do that?
okay. Let's see, I'll start at the top here. We're going to click, oh yeah, that's me. Would you please ask him to repeat what columns A, B, and incidental are for in the Pollard walk sheet? Sorry if I missed this.
Yeah, absolutely. So you can divide your walk, as we were saying a minute ago, into sections, either based on habitat or based on length, and record the data for each section depending on where you are on your route. And again, incidental is when you happen to see a butterfly on your walk that's not in your 5-meter box, but you want to have a total list of everything that you encountered. You would throw that tally mark into the incidental column.
Let's see here. Please clarify distance, time, period, use of the 5-meter box.
So as you're walking forward, doing a Pollard walk, you're just keeping a mental track of approximately what's 5 meters from you. And if you see a butterfly ahead of you, and if it's more than 5 meters, about 16 feet or so, then if you're walking toward it, you might mentally note, hey, there's a buckeye on the ground about 30 feet up. And as you keep walking, you may encounter that buckeye. It might fly into your 5-meter area, or it may still be sitting on the ground when you get there. If so, then you would then mark that on the data sheet as a tally mark within the 5-meter box. If not, you can put it in that incidental. But you're just keeping track essentially of butterflies that you see within a 5-meter forward distance. So not behind you, but out to the sides, out to the front, and up above.
The next question I think it might have answered is there an app or a printable template we use for the Pollard walks? And as I ask you to send me what you've got as a file on email, people can get that from me.
Okay, I'm like I said, happy to send that to you, and people are welcome to contact me as well for any direction, any questions, copy of the data sheet, that kind of thing.
Okay, I was trying to ease you from getting a raft of emails where I was going to try to be the center point, but either way is fine. Okay, let's see the next question. Can you speak to the status of the Palamedes swallowtail? I have red bays that are showing, oops, something's in the way here, just a second.
Yeah, I think I know what they're getting at. So Red Bay and Swamp Bay and along the coast are getting hammered. Their populations are getting significantly, I believe, impacted by the ambrosia beetle?
Yeah.
By the ambrosia beetle that's killing off the trees. And those trees are the primary caterpillar food plant for Palamedes swallowtail. So far, anecdotally, we haven't seen a big plunge or plummet of Palamedes swallowtails. Populations have not been widespread, noticed, or reported that I'm aware of. I'm concerned that it's just a matter of time as we lose more and more of the Red Bay and the Swamp Bay. Now, Palamedes can also use sassafras, which is good. So that gives them a backup, but it's going to be very interesting over the next decade to see what happens to the populations of Persea, of the bay trees, and the swallowtails.
Okay, just not here. Can we report our data now on the Pollard bays? Can we report data as an individual? Whoops, where'd it go?
So the answer to that would be not yet, because the Carolina Butterfly Monitoring Program has not been funded or launched. And so there is no portal on Pollard base at this point for Carolinas. again, once we get the program off the ground, then I would work with the Pollard-based team, get that portal, get our program set up on the website, and that portal would then be available. And then what happens is whenever someone does a survey, they would work with the Carolina director and learn how to use the site and get the data entered and troubleshoot and ask questions and that kind of stuff. So once we get the program up and going, I think there's a lot of potential interest and great data we can collect here in North Carolina.
Okay, I skipped over one here. I didn't mean to. Can a pollard walk be in my own yard?
That is I've gotten that question before, and it's a little tricky to answer because it depends on what your yard looks like and how big it is.
Right.
Pollard walks, ideally about 1 kilometer or 2 kilometers long. They can certainly be longer and they can certainly be shorter. When I was in Montana, I conducted about 14 pollard walks a week. The shortest one was about 300 meters, and the longest one was over 2 miles. So you can certainly set them up at different lengths. So it depends on what your yard, how big your yard is. And you certainly could if you had a fantastic backyard. I spent a lot of the quarantining period of the first year of the pandemic building a pollinator garden in our backyard. It's about 40 feet by 60 feet. And it's been really fun, but it's not big enough for me to really do a pollard walk in, if that makes sense.
Okay, I think this is kind of a question to both of us. May we share the talk with other groups once you send out the recording? I will have a recording, and if it's all right with you, may I share that with anybody that asks for it?
Absolutely, absolutely.
Okay.
And feel free to have anyone who is interested, wants more information, have them contact me.
And I will send this copy of this recording to you, Jeff, just in case you want to pass it on or not
Great.
have the talk, but have a recording for someone. Okay, yeah, and you answered this, I think, but would it be helpful to do the pollard walks and then save the data till the happy day that it gets funded? I think you already answered that.
Yeah, I think it's great. Start a pollard walk as soon as you can, and then just keep those data sheets in a file until the program is launched and off the ground, and then that back data can be entered and be great. From the standpoint of population monitoring, the sooner we can start, the better for sure.
Yeah, and I think the pollard walk today would not bear much fruit.
You are correct about that. Generally speaking, and this is very general, pollard walks run about April 1st to October 1st. Now here in North Carolina, I'm recommending, weather permitting, that they run more like whenever the first warm period in February is, until you get so many freezes that you can't do it. I ran my pollard walk, my last one this year was mid or late November, and the year before was late November, and I hope to start in February. Last year I didn't have weather and schedule that were cooperative enough for me to start until the end of March. But again, ideally once a week, starting as soon as the weather allows, and going as many weeks as you can. But I missed the entire month of June this past year because I was out of the country on a trip. Yeah. And that is okay. So that's one of the great things about this. More data is better, but if you only get 10 of the 30 weeks in, that's all right. That's still good data.
Okay, this isn't exactly a question, kind of a comment, but it might be a question. Charlie wanted to offer that I've made a PDF of butterflies found in the Outer Banks. Contact coastal Dare County using Terry LeGrand's data to make it easier for our citizen science at the Roanoke Island Aquarium to rule out species not seen in our area. My guess is not many people in my area of the state, but if anyone is, I'm happy to share. Just let me know. And that's
Fantastic. And I do know folks at the aquarium that are actively interested in butterflies and have worked with me to get some pollard walks going, and I applaud their efforts.
That's Rachel, it says. I'm guessing it's Rachel Veal. Anyway, here's one from Barbara. Ideally, how long should a walk be?
Yeah, again, good question. It can be as short as a few hundred meters or as long as you're willing to walk and able to walk on a repeated basis. What seems to work out really well, ideally, are 1 or 2 or 3 kilometers. I think that the Duke Forest transect that I set up, excuse me, is, I want to say I keep flip-flopping back and forth in units, I know, but I think it's about 1.5 miles is the transect that I run every week in Duke Forest.
Okay, this is not a question, but I'm sorry that they did it. Sorry, we got hit with power outage in Nashville.
Oh, no.
Okay, do you anticipate funding awards sometime in 2022 or later? Do you know?
I would love to.
You don't know.
Currently, I don't have the proposal in the reviewing agency's hands. So if anyone out there knows of some good foundations or agencies that are looking for proposals like this, please let me know.
Here's a thank you for what you've done. And
thank
I
you.
got Beth Richards, who I know locally, made a couple of contacts from a garden club take part in a workshop, and I will say most certainly myself, I will be contacting not just our members if we get a workshop going, not our members, but all the people I know in my garden clubs and Audubon and everything else.
Wonderful.
And see, Terry, to everyone, since this is a Native Plant Society group, I wonder if there's any value in creating a database of the host plants for each of the butterfly species common to North Carolina. I would say yes, but I would also narrow it down to the coastal plain or wherever you are.
Well, and to some extent, the Natural Heritage Program, Harry LeGrand and Tom Howard, and Harry and Tom update an online publication or a book, even though Harry doesn't say that it is. It's an approximation in its 27th or 28th year. And in that, they have species accounts that list a lot of the plants that are used as nectar and or food sources for the different species of butterflies. In terms of a formalized database, that it would be great to develop that. One of the things I didn't mention, one of the columns on the data sheet that I have is notes or behaviors. And so whenever I see a butterfly nectaring on a plant or ovipositing, laying an egg on a plant, I include that in the data sheet. And so I'll put an N for nectaring, and I'll say Helenium amarum or something like that, depending on what it's nectaring on, or ovipositing on panicum if I see a little grass skipper that's laying an egg on a panic grass type of thing. So if you have the ability to track that data during your walk, fantastic. And the powers that be are working on a new Pollard-based 2.0 that hopefully will get launched by the end of this year or next year, that will have the ability to incorporate other taxonomic groups, including plants, dragonflies, and things like that. So keep track of it. And in terms of building databases, yeah, there's definitely interest.
Okay, there's one that says it's direct to me, but I'm not sure I understand the question. I think it's more for you. Does a Pollard-based team have any suggestions / avenues for funding? I'm not sure understand that question exactly.
I'm not sure either, but like I said, I'm open to ideas.
Could Jeff share his preferred email address? Could you put that in the chat? Would you mind?
I'm happy to.
Okay.
So it's just my name, Jeff Pippin, the number 9 at gmail.com.
Okay.
And it's on, I have that email address also on at the bottom of all my web pages as well.
Okay.
So
All
if
right.
you Google Jeff Pippin Nature, Jeff's Nature pages, that'll take folks to my website and can find the email address there. You may sometimes come across an old duke.edu email. That does not work anymore. I don't have that one. So jeffpippin9 at gmail.com is the best way to get a hold of me.
We got several of them thanking you for the presentation. I'll add to it, Jeff. Thank you so much for this. We hope we will continue with this contact with you, and as this program continues and gets funded, that would be a fantastic thing, I think. I'm thrilled about it.
Well, thank you again for hosting me, and to everybody who had an opportunity to watch and listen, and to the weather for keeping most people in power. I'm sorry that some folks lost their power, but I hope that it doesn't get worse over the course of the day, and I hope all your football teams win as well.
Okay, Jeff.
I know mine was winning at the time that the presentation started, but we'll just leave it at that.
Okay, all right. Well, I'm going ahead and end this, and it will then begin saving it, and I will send it to you, and anybody that asks me for it, I'll send it to them. And usually I can post some of this stuff on the NC Native Plant Society's website, so I may do that if I can. Anyway, thanks again, and hope to be talking to you some more.
Well, thank you, everyone. Stay safe, take care, and have a good 2022.
Okay, bye-bye.